Now, I've told you that the attraction to the microcosm is chemically mediated, and we have, using GCMS, we've been able to find out all the volatiles that this microcosm releases in order to attract wasps. And we've been able to characterize all of them. And I like to think of this as a volatilome. So we have transcriptome, proteome, and now we have a volatilome, which is actually the expression of genes which are actually causing these volatiles to be emitted. Most of them are terpenes and sesquiterpenes, and we've been able to show how the volatilome changes with developmental time. Now, I've told you that there are many hundreds of species of figs. India has about 80 species of figs. Many of them occur in the same place. And if they occur in the same place, and if the wasps are attracted to the figs because of this chemical volatile odor, and I told you that wasps cannot afford to make a mistake. They can't come to the wrong fig, especially if they are pollinators. So we've been asking the question, how are we sure that wasps do not make mistakes? And we've been able to demonstrate that if we look at the volatilome of fig species that occur in the same area, you will find that all their odors are very, very distinctly and separated in multidimensional space, which means that yes, the wasps should be able to differentiate between the odors coming from different figs. More recently, we've been looking at the olfactory receptor genes in figs. And as you can see, if you look at olfactory receptor genes in generalist insects like Apis mellifera or the honeybee, you get about 162 olfactory receptor genes. But in our work with fig wasps, in the pollinator of Ficus racemosa, we get a massive reduction of olfactory receptor genes, which are just 56 in number. And this is a work that is in progress. So, as I've said, we are also looking, we've sequenced the whole genome of the pollinator of Ficus racemosa, and we are now looking at rigorously annotating all parts of, the genome coming, or expression of the genome in different parts of pollinator wasp tissue. And very interestingly, we find that there is a lot of expression also in the ovipositor of the fig wasp, and I will come to that a little later. Okay, just remember that the fig wasp egg laying organ also expresses olfactory receptor genes. Now, in this microcosm, we also have predators outside the microcosm that want to interact with wasps that are entering the microcosm. And here you can see predatory ants trying to feast on the pollinator wasps that are rushing into the microcosm. And we were able to actually demonstrate that ants are able to learn the scent of the figs in the right stages. And this is a learned behavior. So for the first time, we were able to show that the response to fig-associated odors is a learned response by the predatory ants. And this is the first example of eavesdropping, chemical eavesdropping by an ant species on a plant volatile odor. Now, we've also investigated many other organisms within our microcosm, because we have a thriving community of nematodes also in this microcosm. And this association between fig wasps and nematodes is very, very ancient, goes back to about 45 million years. And I have a PhD student who has opened up this whole world of nematodes to us. We now know that we have plant parasitic nematodes, we have animal parasitic nematodes, we have omnivorous nematodes. But what's more important is that these nematodes are very species specific. So they only occur in one species of fig, They don't occur in other ones. They're specific to the fig species. And moreover, they hitchhike. So they get a ride from one fig to another using the female pollinators. And we've been able to show using experiments that the nematodes are able to differentiate between female pollinators and all the other females of the other 6 species of wasps and all the males. So they are very, very, very specific to the sex and to the species of wasp inhabitant. We've looked at the ways in which the nematodes are able to do this, and we think they are using a combination of volatiles coming from the wasps and the volatiles of the cuticular hydrocarbons that are present on the surface of the wasps. Now, hitchhiking nematodes are not always benign, and my student is currently looking at how these nematodes actually affect the mutualism between the wasps and the figs, the pollinating fig wasps and the figs. Okay, so I told you that the only way that the illegitimate fig wasps can enter the microcosm is through the ovipositor. So therefore, we have been looking at the sensory properties of the ovipositor. And we now know that the ovipositor is a marvelous organ. It is as thin as the hair, one human hair. So if you take one of your hair, that is the thickness of the ovipositor. Despite that, you can see that the ovipositor has got all these fantastic tools in order to cut into the microcosm. And we have even measured using very sophisticated equipment the amount of force that the ovipositor has to be able to exert in order to penetrate into the microcosm, and we have measured it in newtons and micronewtons as to what is the force required. I earlier told you that we have established for the first time that the ovipositor of insects is a smelling organ. Before this, everybody thought that only the antennae of insects functions to smell, but we have been able to now actually demonstrate that there are sensory olfactory receptor neurons in the tip of the insect ovipositor, and this is how the fig wasps, the illegitimate fig wasps, that have to find their targets inside the microcosm are actually using CO2 as well as other volatiles that are coming out from the developing wasp larvae. So we've also been looking at chemical footprints that the wasps leave on the surface of the microcosm. Sometimes you will see many wasps aggregating and talking to each other chemically, and we've been able to identify chemical signatures coming from the tiny feet of the wasps. The wasps' feet have glands called tarsal glands which secrete cuticular hydrocarbons, and we've been able to demonstrate that the wasps are leaving their chemical footprints on the surface of the microcosm. So this is all really marvelous stuff, that wasps are talking to each other and informing each other whether they have been there, how many of them have been there, and so on and so forth.